there's a subtle separation in genetics, or in the term gene, and it's into what's called somatic genes and germline genes. Germline genes are genes we inherit from our parents, that we have the day we're conceived, that we carry in every cell in our body, that never change from the day we're conceived to the day we die. They are the genes that dictate all our biological processes, determine what hair color we have, what eye color we have, and they can be altered occasionally. A lot of that alteration is just normal human variation that makes us all different from one another. An extension of that normal variation is abnormal variation, where those germline genes are changed to the point that they can't do what they were designed to do, and they can't produce hair color, and therefore someone doesn't have hair color. They can't produce skin color, and someone has vitiligo. They've lost a skin pigment, or they can't protect us against cancer, and someone has an increased risk of getting cancer. Now, nature being nature considers protecting someone from cancer more important than giving someone hair color, and as a result has lots of backup mechanisms. So our genes in some way are redundant. They cover for each other, and if we've one that's defective, other ones often step in to protect us and do the job that other gene was designed to do in the first place. Genetic screening is a way of identifying people who have an increased risk of disease, and we're talking about cancer, but genetic screening can be used for a whole variety of diseases. And what you're trying to do is you're trying to define somebody by their genetic makeup, and you're trying to define their risk of getting a disease by virtue of the genes that they have inherited from their parents. And what it allows us to do is identify a subset of the population who have an increased risk of developing disease, and then we can care for those people in a specific way that helps to alleviate that risk.
What are the conditions for being genetically screened at the moment, the boxes that a patient has to tick?
So the most important one is family history, and it's a very reliable way of identifying risk, and we often see multiple generations affected by cancers. We see people affected by cancer at earlier ages than we typically expect them to be affected by cancer. We oftentimes see constellations of particular cancers that are associated with particular syndromes. For example, the BRCA genes, most commonly in women, they're associated with breast cancer and ovarian cancer, and the syndrome that a mutation in a BRCA gene causes is called hereditary breast ovarian cancer syndrome. So when I see a patient or a family for a consultation, the first thing we do is we draw out a family tree, and we look for subsequent generations affected. We look for people affected at a young age. We look for those constellations of different cancers that give a clue to a certain syndrome that can be explained by a certain gene that we can potentially test for. There are two BRCA genes, BRCA1 or BRCA1 and BRCA2 or BRCA2. They were both discovered in 1993 and 1994 respectively, and BRCA stands for breast cancer. They are called tumor suppressor genes. So what that means is that their role in life is to suppress the growth of tumors. They protect us against cancer. And when one of them is defective, a person who carries a defect, or another word we use for that defect is mutation in one of those genes, has an increased risk of developing certain cancers. Again, it doesn't mean that person will get cancer, because there are other genes that protect us against cancer. But that person has an increased risk of getting cancer, and as a result has to be cared for in a specific way. P53 is another gene that belongs to the same family of genes as BRCA1 and BRCA2. It's a tumor suppressor gene. It's also known as the gatekeeper of the genome. It's very important in eradicating cells that have very damaged DNA. And if that gene is faulty, there is a risk that those damaged cells can accumulate. And when those damaged cells accumulate, that's what cancer is effectively. And those cells which damage DNA acquire further damage, and it's that further damage that allows them develop the characteristics that we associate with cancer. So a p53 defect, or a person who carries a defect in the p53 gene, confers a high risk of cancer on a person who carries that mutation in the p53 gene.
How likely is it, do you think, we're going to find more and more that genes that you're born with are part of the story?
I think there's a lot more left to discover. I look after a lot of families that clearly have a hereditary predisposition to cancer, and we can't identify it. In fact, the majority of families that we look after in genetics clinics, we can't identify or we can't explain genetically why this is running in the family. I think, I think all cancer occurs because of an unfortunate collision between environment and genetics. And I think the environment still plays a role. We already discussed that not all BRCA carriers get breast cancer. So that either some people have other genes that protect them. So there's, as well as being cancer-causing genes, there's cancer protection genes, and if you have the right combination of the two, you're protected. Or you have to encounter an environmental stimulus. Now, for breast cancer, that's generally estrogen, and there's nothing we can do really about reducing that environmental exposure. But for other cancers, there are environmental exposures that we can reduce. But I think even that environmental exposure, how it then causes cancer, it does so generally through genetically altering cells. So in that aspect, all cancer is genetic, and potentially even that susceptibility to environmental toxins causing cancer is hereditary. And so there might be a lot more about cancer that is hereditary. I mean, even so that this path that a cancer cell goes on, where it starts in, whether it be the bowel or the prostate or the breast, and invades and goes into the bloodstream and travels. To do that, it uses a lot of the genes that are instrumental in embryology, in developing the embryo. And those genes are genes that are inherited, and the cancer then uses them. So it may be that you need certain genes or certain versions of genes that we use in everyday life that the cancer cell then uses to cause problems.